everybody. Welcome back to the OpenStack Psychology 2E Unit 5. My name is Prescott. I'm here at PsychSync, or Succinct Psychology. And last time we talked a good bit about visual pathways, the optic chiasm, photoreceptors, the anatomy of the visual system, all that good stuff. And today we're going to start with the anatomy of the auditory system. So, the ear is divided into three divisions, the pinna and tympanic membrane. So you can see right here, pinna, tympanic membrane, this kind of weird oval-shaped thing. We have the middle ear, which are the three ossicles, or the malleus, incus, and stapes. You can see right here. I'll zoom in a bit. Malleus, incus, stapes. Then we have the inner ear, which is the cochlea, right here, and the basilar membrane and the hair cells. So, going on with that, we have a more in-depth illustration of the anatomy of the auditory systems, and it again highlights what makes up the external ear, the middle ear, and the inner ear. So, moving on, we have auditory transduction. So, sound waves travel along the auditory canal and strike the tympanic membrane, causing it to vibrate. The vibration causes the three ossicles to move. This presses the stapes into the oval window of the cochlea. The fluid inside the cochlea begins to move, stimulating the hair cells, sensory receptors for sound, which become activated. The hair cells generate neural impulses that travel along the auditory nerve to the brain. Auditory information is sent to the inferior colliculus, then the medial geniculate nucleus of the thalamus, and finally to the auditory cortex in the temporal lobe for processing. So that's auditory transduction. Pretty complicated there, isn't it? Next, we have pitch perception. So how does the auditory system differentiate among various pitches? So we have two theories here. We have temporal theory, which is frequency is coded by the activity level of a sensory neuron, but there's a problem, and that is the frequency of action potentials cannot account for the entire range that we are able to hear. There's a point at which a cell simply cannot fire any faster. Next, we have place theory. So different portions of the basilar membrane are sensitive to sounds of different frequencies. The base responds to high frequencies, and the tip responds to low frequencies. Both theories explain different aspects of pitch perception. So the rate of action potentials applies up to about 4,000 hertz, but higher frequencies can only be encoded using place cues. All right, next we have sound localization. So localizing sound involves the use of two cues. Monaural cues, I almost said monaural, so it means one ear, and each ear interacts with incoming sound waves differently. And then we have binaural cues, which is two ears, and provide information on the location of sound along a horizontal axis, so flat axis. It relies on differences in patterns of vibration. So we have interoral level difference, which is sound coming from one side of the body is more intense at the closest ear because of the attenuation of the sound wave as it passes through the head. And then we have interoral timing difference, which is small difference in the time at which a given sound wave arrives at each ear. And so these two differences are depicted here. So in this figure, jets are flying overhead. So the level difference, the closest ear is going to be more, the sound's going to be more intense in that ear that's closer to the origin of the sound. And then the timing difference, that ear that is closest is going to receive the sound before the ear that is facing away from the sound. Next, we have hearing loss. So deafness is the partial or complete inability to hear, and congenital deafness is deafness from birth. Then we have conductive hearing loss, which is associated with a failure in the vibration of the eardrum and or movement of the ossicles. Can often be dealt with using hearing aids, which amplify incoming sound waves to make vibration of the eardrum and movement of the ossicles more likely to occur. And it can be caused by age, genetic predisposition, environmental effects, exposure to extreme noise, certain illnesses, or damage due to toxins. So sensory neural hearing loss is the failure to transmit neural signals from the cochlea to the brain. And it can be caused by Meniere's, I might be saying that wrong, disease, which results in a degeneration of inner ear structures, which obviously is not good. Your ear is kind of important. So here we have conductive hearing loss. So environmental factors that can lead to conductive hearing loss include regular exposure to loud music or construction equipment. So, figure A, loud music, figure B, construction zone sounds. If you've not been on a construction zone before, it can get very, very loud and to the point where it's painful. Construction workers, obviously, because of that, are at risk for this type of hearing loss. Going on from hearing, we have taste or gustation. Research demonstrates that we have about 6 groupings of taste. These are sweet, salty, sour, bitter, umami. It's associated with a taste for monosodium glutamate. And then the 6th one is some research suggests we possess a taste for the fatty content of food. Taste buds are groupings of taste receptor cells with hair-like extensions that protrude into the central pore of the taste bud, and they have a half-life cycle of 10 days to 2 weeks. Now, transduction of taste is taste molecules bind to receptors and cause chemical changes within the sensory cell. These changes result in neural impulses being sent to the brain. So here we have visualization. So taste buds are composed of a number of individual taste receptor cells, as you can see here, that transmit information to the nerves. So tongue surface, taste core, so you're eating something, that's where your taste receptor cells detect the taste and it sends nerves to transmit information to the brain. So this micrograph, figure B, shows a close-up view of the tongue surface. So this is a little cartoon drawing, but this is what it actually looks like, which looks kind of gross, if I'm being honest. Next, we have smell or olfaction. So olfactory receptor cells contain small hair-like extensions which serve as the site for odor molecules to interact with chemical receptors located on these extensions, located in a mucous membrane at the top of the nose. So here we have the olfactory receptors, there's the nasal cavity, so inside your nose, and there's the olfactory bulb. So, transduction of smell. Odor molecules bind to receptors. Chemical changes cause signals to be sent to the olfactory bulb, right there, where the olfactory nerves begin. Information is sent to the limbic system and primary olfactory cortex. Now, pheromones are chemical messages sent by another individual. Many species respond to pheromones sent by another individual, and they usually communicate information about the reproductive status of a potential mate. Next, just going down a list of senses, we have touch. So there are many types of sensory receptors located in the skin, each attuned to specific touch-related stimuli. So we have Meissner's corpuscles, I don't know how to say that properly, respond to pressure and lower frequency vibrations. We have Pacinian corpuscles, which detect transient pressure and higher frequency vibrations, so Meissner's and Pacinian. Then we have Merkel's discs, right here, which respond to light pressure, and Ruffini corpuscles, which detect stretch. And when I first looked at this, I was like, oh yeah, it kind of looks like something that you would pull on and stretch apart like a plastic toy or something. Or a rubber toy, I should say. Next, we have thermoception and nociception. So as well as receptors located in the skin, there are a number of free nerve endings that have sensory functions. So thermoception is temperature perception, temperature, like a thermometer. Nociception is the sensory signal indicating potential harm and maybe pain. This type of sensory information travels up the spinal cord directly to the brain, specifically the medulla, thalamus, and the somatosensory cortex. Next, we have pain perception. So pain perception is important because it motivates us to remove ourselves from the cause of injury. So inflammatory pain, like arthritis, signals some type of tissue damage. And then you have neuropathic pain, which is caused by damage to neurons of either the peripheral or the central nervous system. Now, congenital insensitivity to pain is a rare genetic disorder in which the individual is born without the ability to feel pain. They can detect differences in temperature and pressure, but they cannot experience pain. And individuals are at greater risk of injury and have shorter life expectancies when they have congenital insensitivity to pain. And I actually remember one of my, learning about this with one of my professors showed a video of this guy who had congenital insensitivity to pain, and how he would, fall, or like he was just constantly getting bruised or hurt because he couldn't feel the pain, and so he wouldn't know that anything was wrong. So it's not good. So next we have the vestibular sense, which is our ability to maintain body posture. And this is weird. It's kind of cut off, so I'll just go on to the next slide because I'm only getting half of the words. I don't want it to be broken. But here you have vestibular sense, like it's in your ear, because you can see the cochlea, basilar membranes, the inner ear. So the vestibular sense, the maculae are specialized for sensing, you can see right here, maculae, for sensing linear acceleration, such as when gravity acts on the tilting head, or if the head starts moving in a straight line. So here you have head tilted forward, and here you have a head upright. Difference in inertia between the hair cell stereocilia and otolithic membrane in which they are embedded leads to a shearing force that causes the stereocilia to bend in the direction of the linear acceleration. So here you have the head tilted forward. It's causing the stereocilia to bend forward because of the force of gravity, whereas when your head is upright, like mine is right now, they stay level. So we will finish up Unit 5 in the next video. We will go on to proprioception and kinesthesia, and then we will finish up Unit 5. But I hope you guys enjoyed. Again, my name is Prescott here at PsychSync, Sync Psychology, and I hope to see you in the next video where we finish up Unit 5 of the OpenStack Psychology 2E lecture slides. Thank you, and have a great day.